All right, sorry about the delay, folks. We'll get underway, and we'll try to make sure that we still are able to finish on time. Apologies to those who are listening at home. Unfortunately, it's going to be recorded, but it's actually not going out live today. There are some issues with the microphone. So I wanted to go back over major depressive disorder. So there's a couple of things that I really want to get across. I have a lot of different emails and communications with various individuals in the class, which I think is great because that's part of the reason I gave that talk, is to get you to think about mental health. What I'm trying to convince you to also think about is don't make a self-diagnosis. You're going to see things up here that probably describe you, but you are not a clinician. I am not a clinician. And you need to actually get a medical diagnosis for a variety of reasons. And some of those reasons are coming up in the next few slides. But a clinician is going to ask you if you have had at least once a day of any of these different types of conditions, whether it's a persistent feeling of sadness or hopelessness, if you just don't enjoy things that you used to enjoy before, if you have a change in appetite, a change in your sleep cycles. All of these represent, and you don't have to memorize this, all of these represent changes in different brain circuits, and there are about 3 or 4 brain circuits that are clearly defined by each of these different things, whether it's a sleep-related circuit, and circadian rhythm-mediated circuit, whether it is the VTA nucleus accumbens, potentially other areas of the brain that are being activated. All of these suggest that it's not one system in your brain that's being changed by major depressive disorder. There are many circuits that are actually being altered by major depression that cause all of these different types of things. Ultimately, though, you're going to, based on those different questions related to figuring out which of these circuits are being affected in depression, after you fill out something like a PHQ-9 questionnaire where a clinician is going to be asking you these questions, you're going to have a number assigned to you. So if you thought that you were just a number here at U of T, you're also just a number when you visit your psychologist. So I was also assigned a number as well many years ago. But sometimes it actually is really useful. So if you've gone through this whole process and it turns out that you have really, really severe symptoms, then you are on a different sort of protocol than someone who might have moderate symptoms. I know it sounds terrible, but because the healthcare system is overburdened, that's the way that these things tend to work. Again, this slide not being important for you to know in any detail. But I do want you to think about this. We know, for example, that there is a biology behind it. That's ultimately what I'm here to talk to you about today, what you need to know for your upcoming term tests as well. And one of the reasons why I want you to go and visit a clinician, if you feel that you have any of these different depressive or anxiety-related disorders, is they have to be able to rule out that it's not hormonal. Because hormonal changes can actually mimic having depression. And hormonal changes actually don't really respond well to antidepressant medication or therapy. So for example, we know that in individuals who are depressed, there usually are changes in different glucocorticoids. And these glucocorticoids, cortisol for example, when we look at an animal model, if we were to give high levels of glucocorticoids, we were to inject cortisol, corticosterone, or other glucocorticoids directly into an animal that wasn't normally depressed, one of the things that we would see is that after a while, that the high levels of corticosteroids, these glucocorticoids that were being injected, would ultimately lead into the animal being less mobile. It wouldn't move around as much. It would also have less motivation. So if we had an operant conditioning task that was related to motivation, it wouldn't respond well to that. So glucocorticoids definitely do affect one particular circuit, probably the motivational circuit, probably the motor circuit. Again, it's not moving as well. But it's a good model system to tell us that in some cases where we're looking at depression, the reason why we need to look at a clinical diagnosis is because your clinician can actually take a blood sample. They can actually see if your glucocorticoid levels are outside the normal range, and then there might be other treatments that you might be able to get there as well. So the animal model is one I'd like you to make sure that you know. There's some other points on here. So there's a lot of evidence anecdotal evidence, but people like using this type of anecdotal evidence to suggest that changes in hormones actually result in different forms of depression. For example, birth and postpartum depression are often associated with changes in various hormones. If you look at when, for example, individuals who have this have depression-associated changes in mood that are often associated by the menstrual cycle as well. So we know that there might be some hormonal changes that accompany these menstrual cycles. And if you look at this from, again, a purely anecdotal point of view, but this is actually in many of the reviews that we might be reading up on related to depression, when we look at the period of time that you are most vulnerable, the ages of 15 to 44 on average, median age is around 30, this is actually the point in time where you are beginning the menstrual cycle and you are ending the menstrual cycle. So it suggests that their hormonal changes for some individuals actually might play a role in different forms of depression. And again, purely anecdotal. People do research this. The research behind, for example, hormonal-related depression, especially another reason why we're interested in this, females are affected much more than males with depression as well. So again, I don't need you to memorize this list. It's just to give you a different overview of this. If you were to look at the current treatments, and this is really sad and this is really unfortunate, but the first line and the first thing, if you are going to your family doctor, who's not a registered psychologist nor a psychiatrist, you're often just prescribed antidepressant medications based on a brief chat with your family doctor. And these antidepressant medications, they run in a whole bunch of different flavors. So selective serotonin reuptake inhibitors that I'll talk to you about later, very, very commonly given. We still use tricyclic antidepressants or TCAs, and monoamine oxidase inhibitors or MAOA, MAOB inhibitors are still used in some cases as well. But this is the first line of defense. I'm going to try to convince you at the end of today's lecture that sometimes it works really, really well, but the majority of times it doesn't work well for the vast or for a large population. A larger population than we would want to be able to help. But there are other forms of therapy like psychotherapy, talk therapy, like cognitive therapy, behavioral therapy, interpersonal therapy, or even family-mediated therapy, and then, of course, different combinations. I can tell you also that for some individuals who have very, very bad forms of depression, that ECT or electroconvulsive therapy is given. You're usually sedated, and then you're shocked. You're given a shock. It doesn't really cause you pain because you are sedated, but this is still used. It's still given, for example, in places like CAMH. It's not given very often, but if you are treatment resistant, and you have like a score that is suggesting that you are putting yourself at harm, then they will try other methods to make sure that you're on the road to wellness and recovery. I'll be talking very briefly about deep brain stimulation, and of course hospitalization is always an option that should be considered as well. But let me go back and talk to you a little bit about the biology. We know that there are circuits that are being affected, things that probably involve circuits around the hypothalamus that are part of the basic drives. Those are being affected. If you're not eating and sleeping correctly, that means that the hypothalamic circuits that are involved in eating and sleeping are being affected. We know that the prefrontal cortex could also be affected because cognitive disturbances, where you keep thinking about the same thing over and over, where you feel worthless and you feel like you can't make up your mind about things when you have depression. If you have persistent thoughts of suicide, for example, these cognitive disturbances suggest that the prefrontal circuits might also be important as well. We know reward centers are also going to be affected, which means that midbrain structures that ultimately feed into some of the hypothalamic circuits involving appetite, eating, etc., are all interconnected together. This suggests for me that we should, as neurobiologists, be very interested in this. All the things that you learn in physiology, the things that you learned in your neuroscience courses, or other health and disease courses are ones that you should be using to the best of your knowledge. So again, just to point out that when we look at these 9 different categories that are related to changes in your behavior, whether it's physiological changes or psychological changes in the way you think or perceive things, these all kind of relate back to different neuronal circuits. So, for example, I'm going to use this term on your tests, and I wanted everyone to be on the same page. Your inability to enjoy anything, this anhedonia that I'm going to be talking to you about, this anhedonia probably involves circuits that involve the BTA and the nucleus accumbens. And I want you to be aware that if I mention things like anhedonia, if you haven't heard it before in psychology or you haven't heard it before in your physiology classes, just to be on the same page that this is that inability to enjoy things that you used to enjoy. So your inability to move in the morning, your inability to get out of bed in the morning, the inability to actually want to do something are probably psychomotor effects. And again, this also probably ties in to other circuits. So I want to kind of go back and think about depression, anxiety, because they're kind of related. And I want to start thinking about how we look at this from a neurobiological perspective. I know many of you are taking psychology, and many of you will learn very similar things. But I actually want to look at it from a slightly different perspective that we're going to move into in the next lecture after the term test. So we're going to start thinking about, okay, so if these circuits are affected, and if the current way that we look at depression, anxiety, other mood disorders is maybe not correct, how should we actually go about rethinking this as a biological circuit-related disorder, and how do things like exercise play a role in trying to reset this whole system? Or does it reset this whole system, and how might we understand it from a biological perspective? So there are a couple of things on this slide that I want you to think about a little bit. So we know very little about depression, and I mentioned this to you before, that most of this actually appears to be idiopathic. We don't know the cause, so it's not unlike Parkinson's. We don't really have a genetic cause for depression. We know that there might be a genetic risk for depression, but there's no genetic cause for depression. We don't know many of the risk factors. We know that stress is probably the biggest one that will be a trigger for depression. We know that there are some hormones that I mentioned briefly, glucocorticoids, that I'm going to come back and talk to you about a little bit more in the next few slides. And we know that some cancers are also capable of inducing depression. So I'm not talking about the treatment causing depression. I'm talking about if you have pancreatic adenocarcinoma, before you get the diagnosis of pancreatic adenocarcinoma or a breast tumor, for a variety of reasons that we don't fully understand, those individuals before the diagnosis of cancer will also have depression as well. So there's a lot of biology, there's a lot of signaling from different parts of your body that will go and affect this. We know that if we were to inject you with drugs like isotretinoin or interferon alpha, this can, in some individuals, actually trigger depression as well. So you'll be perfectly fine. You'll get an overdose of isotretinoin or interferon alpha or interferon gamma, and then this will trigger depression in some individuals. But again, it suggests that there's a biology, there's a target, and we know that that's an important consideration when we're looking at the biology of depression. The biggest drawback for us right now, we don't have a gene, and the way that we like to work in biology is we have a target gene, which then causes us to either look at its expression levels, like if we knock it out, what happens? Does the depression go away? If we overexpress it, do we get more depression or protection from depression? But right now we don't really have a target gene, and therefore we don't have a model organism associated with depression. So we're really kind of stuck with trying to make our best guess. And some of our best guesses go back to what I told you before. There's some anecdotal evidence that hormones do play a large part, and that some physicians, some clinicians, and some scientists think that up to 20% of diagnosed depression is actually mediated by some sort of hormonal balance, which again is a strong indicator for you not to self-diagnose, to go in and see not just your family doctor, but also like a psychologist or a psychiatrist. So one of the diseases that I'd like you to make sure that you are aware of is Hashimoto's disease. And Hashimoto's disease is where we have an autoimmune disorder that actually causes a loss of thyroid gland-related cells. So the immune system starts to attack the individual's own thyroid cells. The thyroid gland gets destroyed in this, and as a result of this, this causes hypothyroidism. So this hypothyroidism that results from this autoimmune disorder that we're calling Hashimoto's disease will actually cause this individual to start feeling tired all the time. They don't want to move. They feel very sluggish when they do move. It's often associated, because it's a thyroid-related condition, to an increased sensitivity to cold. Not important to us, but for an endocrinologist, really important. There are changes to the skin. There are changes to the overall appearance. And most interesting to us in neurobiology, if we start losing thyroid hormone and we have hypothyroidism, this actually causes changes in increased anxiety. It will cause problems in your sleep. It will also cause weight-related changes. This individual is actually experiencing an increase in weight, but it can also go the other way. The vast majority is an increase in weight, but there are some cases where weight-related changes can actually cause a loss in weight. But these are all indicative of things that resemble the characteristic features of depression that I mentioned to you a few slides ago. This also, by the way, again, purely anecdotal, but it's an interesting anecdote, affects women much more than it affects men. Also, it results in things that look a lot like depression. So, if we replace the thyroid hormone and we can supplement thyroid hormone, actually all of these symptoms disappear in this individual. Anxiety goes away, depression goes away, changes in sleep goes away, the changes in the structure of the face go away, all of these things go back to normal. So this is one form of where we get a biological cause related to hormones that will cause depression as well as anxiety. So that's case one. I've already mentioned to you about glucocorticoids. So there's this other condition which is known as Cushing's syndrome. And in Cushing's syndrome, there's another hormone that's involved, very much like the glucocorticoids that I mentioned to you earlier. So glucocorticoids, when you start overexpressing glucocorticoids, so if I inject high levels of glucocorticoids, not only does it cause a loss of operant motivational conditioning in those animals, but it will also cause a decrease in dopamine within the brain in those animals. And one of the interesting things about Cushing's syndrome is that it also has a sequela, or a constellation of features that actually resembles depression. In Cushing's syndrome, where we have too much of this glucocorticoid, they will also fall into depression as well. So again, we're looking at these glucocorticoids, things like corticosterone or cortisol. Hopefully this is a really simplistic review for you of the neurophysiology that you learned earlier, but just as a review. So for example, within the hypothalamus will release corticotropin-releasing hormone, or CRH, and normally corticotropin-releasing hormone released from the hypothalamus will travel to the pituitary gland, and the pituitary gland, in response to this hormone, will also release ACTH from itself. So the pituitary gland now will release ACTH, the adrenocorticotropin-releasing hormone, which will then circulate throughout the blood and then target the adrenal glands. And these adrenal glands that are sitting on top of the kidney, they're suprarenal, will then, in response to elevated levels of ACTH, release cortisol into the bloodstream. If you have stress, what is the hormone that goes up in your bloodstream? Cortisol is that hormone. So stress, cortisol, Cushing's syndrome, cortisol. Does it have the same sort of phenomenology that we are looking for as well? So one of the things that we see in these individuals, and here's a slightly diagrammatic view of this. Some of this will make sense for depression. Some of it makes more sense just as a purely hormonal issue. So the skin ulcerations, the purpura, for example, you don't need to worry about these, but the ones that I've outlined for you, especially like an increase in weight, especially in the increase in weight redistribution that is associated with the midline, so around the waist, as well as on the back, which is a very characteristic buffalo hump, which was on this previous slide. This is a good sign of Cushing's disease, which means that the fat is being redistributed to other parts of your body as you actually increase the levels of cortisol. This is what Cushing's disease is. Your cortisol levels are always elevated. It's like feeling stressed out all the time. And again, there are all sorts of emotional disturbances. These individuals have depression, anxiety, they are very emotional all the time, especially when cortisol is high. And one of the things that is often done is if you go to the doctor and they suspect for a variety of reasons that your changes in eating haven't changed, but your weight has changed, that if they suspect things like Cushing's, they will give you what is known as a dexamethasone test. They will inject dexamethasone, and they will see if your cortisol levels go up or they stay the same. Now, I'm not going to go into all of the details. You might see dexamethasone as a test or an indicator to look for Cushing syndrome, which is related to increased glucocorticoid levels, but I'm not going to get into the clinical relevance of the dexamethasone test. Overall, though, I mentioned to you earlier, this HPA, the hypothalamic circuit, which involves sleeping and eating, is really important. And in this case, the glucocorticoids actually drive the circuit in a slightly different way, resulting in changes in emotion, changes in weight distribution. There are also sleep disturbances, again, suggesting that the circuit, much like the circuit that we see in depression, is being affected. And we know that this is a system that is being overstimulated. There's way too much glucocorticoid out there. It's like the person is stressed out all the time, and as a result of this actually causes weight changes, changes in weight distribution, and fat deposition in places that you normally wouldn't expect. So this gets into this really interesting drug, which I'd like you to make sure that you know, and we'll go by the name RU486. We'll see RU486 quite frequently in neurobiology, several different times throughout this course. And what I want you to make sure that you know is that RU486 is a glucocorticoid or a cortisol receptor blocker. So glucocorticoid receptor blocker or a cortisol receptor blocker. And if we block glucocorticoid from binding to its normal receptor, then one of the things that we see happening is that in some cases, even in depression, not just in Cushing syndrome, we will actually start to see that this can be used as a non-traditional antidepressant, which is by short form of ADM, antidepressant medication therapy. So normally that's not on that list, but for some individuals that maybe don't even have the symptoms of Cushing's, if we give them RU486, even if they don't have Cushing's, we can actually help them with severe depression, especially if they also have psychosis associated with that depression. So again, it begs the question, why does blocking a glucocorticoid receptor block depression if that's not the primary cause? It's not the way that antidepressant medications are supposed to work, and yet for some individuals, it is actually very, very potent. All of this kind of suggests, if we're looking at the basic foundation of neuroscience, that somehow neurons are responsible for this. Neurons in a circuit are responsible for this. If we look at the basic neurobiological sort of doctrine, that means that in a circuit, if we're changing a circuit, usually we're looking at a change between neurons. And the way that neurons communicate are through these neurotransmitters. So this strongly suggested that there is a change in neurotransmitter levels in individuals that have depression. And this is the most common theory that is out there. It's still the dominant theory that's out there related to depression. Many of you have taken courses, whether it's psychology or biology, related to depression, and you've all heard that there's a change in your neurotransmitter levels. That's why you take selective serotonin reuptake inhibitors or selective serotonin and noradrenaline reuptake inhibitors. You want to get the neurotransmitter levels back up. There is no doubt that the circuits that we're interested in that are being affected all make use of these monoamines, which are also referred to as biogenic amines. And again, you'll see later, catecholamines are part of these. And many of these play a very pivotal role in different types of behavior related to motivation, movement, pleasure, anhedonia. All of these different biogenic amines are thought to be really important. I'm going to show you some initial experiments using various animal models that manipulate these neurotransmitters that are bioamines. So things like dopamine, serotonin, noradrenaline, that if we manipulate them by injecting different compounds, we reduce or increase these levels, we will be able to induce things that look a lot like depression, which means that neurotransmitters might play a role. So substance abusers also show signs of depression, and they're also stimulating like the dopamine pathways, serotonin pathways, noradrenaline pathways. All of these come into play for individuals with substance abuse, often meaning that neurotransmitters are changed in those individuals with depression who are substance abusers as well. So PET scans also indicate that we have changes when we take a look at the overall levels of specific neurotransmitters like dopamine. So in a depressed individual, the PET scan might look very, very low compared to an individual who's not depressed, suggesting that if we're following the release of neurotransmitters using positron emission tomography, that there's a change in the overall level of release of these neurotransmitters. So let me take you through the dominant theory. This is the neurotransmitter, neurochemical hypothesis of depression. How many of you have heard of this theory related to depression? Honestly, really? That's it? Like 5 people in this entire class? You've never heard that when you have depression, neurotransmitter levels change? Oh, okay, thank you. That's right. Awesome. So when we take a look at this, there's a lot of evidence. So if you were to go all the way back, so it's one of those things that I actually don't want you to go too nuts thinking about, but neuroscience is actually in what we think is like the third golden age. So the first golden age was actually in the 1950s. In the 1950s, there were so many new exciting discoveries that were going on about the brain, neurochemicals, etc. So in the 1950s, there was this huge explosion of our understanding of the neurochemical basis of neurotransmission. We started to figure out different neurotransmitters. We got different drugs. Drug companies were coming out with new drugs that could be injected that would block neurochemicals from being released, etc. So we know that experimental evidence from rats in the 1950s suggested that pathways were involved. We knew, for example, the midline, the midbrain structures were really important for motivation and the release of dopamine. We knew that only in the 1950s. We know that work that was done by Wilder, Penfeld, and others in the 1950s and other neurosurgeons in the 1950s demonstrated that certain anatomical pathways were related to pleasure. So if they were anywhere near the midline in a human, and they started stimulating because they were mapping out the human brain in the 1950s and our understanding of different structures, if we were to place a stimulating electrode during neurosurgical procedures related to epilepsy in the 1950s, if Walter and Penfeld stuck something near the mesolimbic centers and turned on the electrode, the person would say, "Wow, that feels really good. Can you keep stimulating me there? Keep going. Raise it higher. I like it." So we know that these midline structures, very similar to the animal structures that were also identified here at the same time, were probably involved in the release of different neurochemicals. One of these substances actually was really important. This is a substance that was found in the 1950s by one of these drug companies. It's a drug that I'd like you to make sure that you know. And reserpine was originally used as an antihypertensive. So it was designed as a drug by drug companies to actually reduce blood pressure. So that's what an antihypertensive was. It was great because it actually works. So it actually reduces the levels of noradrenaline. So it works as an antidepressant. it works as an antihypertensive, and these people were taking this, their blood pressure was going down because it was reducing the overall levels of noradrenaline. Sounds like a great idea. You go back, for example, to your classic physiology, and you know that adrenaline and noradrenaline release will actually stimulate the heart, increasing blood pressure, and it was a good idea to reduce adrenaline levels that would decrease heart activity, make the hypertension, high blood pressure go away. The problem was people who were getting better because their blood pressure went down actually started to develop a lot of these mental health issues. They actually became depressed. Enough of them became depressed that they started to think about suicide and other things. And that got people to think, how is it that this drug that reduces noradrenaline is actually causing individuals to actually become depressed? So you deplete noradrenaline, does it perhaps deplete noradrenaline in the brain? These are not supposed to be individuals who are depressed. So again, this reserpine actually gave some clue that maybe monoamines, like noradrenaline, might be the root cause of what is involved in depression. We know that other classes of drugs that were, again, for a variety of reasons that I'm not going to get into, drugs like iproniazid and imipramine, that were not originally developed for antidepressant. These were actually also developed for controlling blood pressure, etc. These were also shown to actually have good antidepressant effects. So again, these drugs, and I won't get into all of the details, these drugs also are part of the noradrenergic pathway. They change the noradrenergic pathway. They will actually do the opposite of increasing the levels of noradrenaline. And when they were given to individuals who had a variety of different conditions, unexpectedly, when they were given these drugs, imipramine and iproniazid, when they were asked by their doctors, like, how are you doing today? Actually, I feel pretty good. I feel much better than I did before. And they were thought to actually work by increasing noradrenaline in the brain, and these were thought to be antidepressants. So we have one drug, this is the reserpine, that reduces monoamines, causing depression, and we have other drugs that increase noradrenaline that make people feel better. So wouldn't the logical thing be that noradrenaline or monoamines, changing the levels, will make you depressed or make you feel better? And that was the thought in the 1950s, that by these observations, this should be the logic behind the changes that we see in the brain. So we now know, or not now, but in the 1960s, there was further compelling evidence to suggest that if you also had drugs that would enhance serotonin, not just noradrenaline, but also enhance serotonin and noradrenaline, you could actually make better antidepressant effects. And so it was thought that serotonin and noradrenaline somehow have to work together to actually make depression or make you come out of depression as an antidepressant medication. So this is sort of the foundations of where we're at today in 2017, thinking still about this neurochemical hypothesis. There's a change in neurotransmitters. Might be noradrenaline, might be serotonin, but something is wrong in your brain, and that's what we have to be able to fix. We have to give you drugs that will increase the level of these different substances. So again, just a couple of other things. If you've gone through the mock test, you'll know that this is something I do want you to make sure that you know. And if you were to take a look at these catecholamines, and the catecholamines are a broad class that include adrenaline, noradrenaline, etc. So within the brain, we know that noradrenaline or norepinephrine, it's the same interchangeable term that I'm going to use, is actually produced in discrete and very, very specific regions within the brain. We know that there are two major regions. The cell bodies actually sit in what is known as the locus coeruleus. Do you remember the locus coeruleus by any chance? Do you remember us talking about the locus coeruleus? The blue neurons, the blue group of neurons? So we talked about this as a pigmented group of neurons when we talked about the Parkinson's disease. And this locus coeruleus is the group of neurons that produces noradrenaline. And then there's the group of neurons known as the lateral tegmental neurons. Both of these groups produce 99% of the noradrenaline that occurs within the brain. And these two small discrete regions, the locus coeruleus being down here, and you can't really see the lateral tegmental neurons, but these small group of neurons send their projections to the cerebellum, down the spinal cord, and they will release norepinephrine at all of these different areas. So even though noradrenaline is produced in a very small group of neurons, it has a huge effect because norepinephrine is sent along the dorsal bundle and it gets released from the dorsal bundle. And this is that dorsal adrenergic bundle, the medial forebrain bundle, etc. So a couple of other things that I wanted you to be aware of. Why did people think that this might be a neurotransmitter that's involved in depression? So remember, we have problems with sleep-wake cycles. So noradrenaline plays a very key role in sleep-wake cycles. Again, I don't need to go back and remind you of things that you've learned about in other physiology courses. Suffice it to say, noradrenaline plays a key role in sleep-wake cycles. We know it's also important in vigilance, alertness. It's involved in mediating and modulating the reward and reinforcement of reward. And this other really interesting anecdotal fact is if I have a stroke here, and it's a very, very selective stroke, the blood flow in this region of the brain is actually very selective. You can have a stroke right near the dorsal adrenergic bundle, and this anterior stroke actually results in a very interesting phenomenon. You damage only this dorsal adrenergic bundle if you have an anterior stroke in the dorsal adrenergic bundle. The only side effect to this, there's no loss of speech, there's no motor coordination deficits, but you become depressed. And this lack of having noradrenaline being released at this dorsal adrenergic bundle is actually another thing that many clinicians bring up as catecholamines playing a role in the biogenesis of depression. So I think I have a couple of more slides to get through, and I'm going to try to spend about a few more minutes on it. We will take a break, and we'll have our normal movement break. But let me take you through what you need to know on this particular diagram. I don't need you to memorize all of these things. We're going to come back and talk about some of these later, but I wanted you to have an overview. We have spent a lot of time and money, a lot of research that has gone into the development of drugs that will overall change the level of neurotransmitters here at the level of the synapse. We will have things that will affect the MAO system, the monoamine oxidase system, that is often associated with the mitochondria. And these MAO systems are important in regulating the breakdown of different types of neurotransmitters like dopamine, noradrenaline, etc. And we also have these extracellular forms of MAO or monoamine oxidases as well. And all of these drugs can target them because they normally break down neurotransmitters. But there's this other class that actually cause the blockade. It stops this from working. You stop taking up the 5-HT or serotonin back into the nerve terminals. This is going to be the controversy that we're going to look at as a group and try to understand this controversy. This controversy, by the way, has raised its head again in the last 2 weeks. Hello, at the back. I don't know, I get random people coming to the back, smiling, waving, and running away, giggling. That happens, I guess. This class that we're going to be talking about, the SSRIs, has actually been the biggest source of controversy in pharmacology and neuroscience in the last 20 years. And we're going to go through that particular controversy when we come back from the term test. But I wanted you to have an overview of the different types of drugs that are being developed for this. You don't have to learn this list. Some of you might know these because you might have heard of like sertraline or Zoloft, etc. All of these come with side effects. And the biggest sort of side effect that I wanted you to be aware of, and especially for those of you who are in pharmacology or toxicology, these are great drugs. They work. They work for some individuals. The problem is they now all have like a black box label or a black label on them. And this is because of things that were found, especially in teenagers who are taking these drugs and these medications for depression. It will actually cause an interesting phenomenon where taking these antidepressant medications actually increase their suicidality and the intention of suicidality and ideation. And again, the research behind that isn't fully clear, but it is a well-known fact. And it's one of the reasons why there is that sort of label warning on this. We don't understand this system enough in the developing brain, in other words. One of you will have to be brilliant enough to take on this task of trying to figure out, why do young people taking antidepressant meds actually increase the likelihood of committing suicide or thinking about suicide if it should be helping them get better? So I'm going to take you through this one last slide, and then we'll take a break. I want you to know this slide. I want you to know this slide and think about this slide. And when you're thinking about this slide, I want you to think about me, because I'm that self-centered. So I ask my students, and I'm going to ask the same group, if you have the opportunity while you're young and while you're healthy, get a nice picture of your brain. Because everyone deserves to have a nice picture of your brain while it's nice and healthy. I did not have a nice picture of my brain before I had depression. And it's something that I really regret to this day. I have a picture of my brain for the last 6 years. I've taken one every year right before the start of school. I go in, I get a complete physical before the start of school, and I have 6 wonderful MRIs. But I don't have one before I had depression. I don't have one during depression. And I really regret not doing this. Because I would really like to know, for example, like what was the size of the gray matter before I had depression? Because I want you to think about this. When you are undergoing depression, these are the changes that are often associated with depression. We have changes with less gray matter. So the cortex, for example, we have less of that. We also not just affect the gray matter, we also have a reduction in the white matter. So not just the cell bodies, but also the axons are disappearing. The myelinated axons are disappearing in depression. So we're having a loss of tissue. We have less glia. So it's not just neurons, it's also glia are also being affected in areas that involve learning and memory, so the hippocampus, as well as in the prefrontal cortex. This is often thought, maybe this is why you have things like worthlessness as well as guilt. So the easiest thing for you to do as a neurobiologist, if you're thinking simplistically, is, okay, depression, you feel sad, things are shutting down, it must mean that the entire brain is shrinking, shutting down. Maybe. But there's some other interesting things that are also going on. Some parts are. The gray matter, white matter is, but the anterior cingulate cortex, the area of the brain that's involved in error-related signaling, actually has a resting level that's higher in most depressed individuals. So this kind of goes against that trend. So overall, the brain is shrinking, it's going down in size, especially in the prefrontal cortex and in the hippocampus, but the anterior cingulate is actually going up in activity. We know that the amygdala is also going up in activity. It's hyperactive, it's more active, but also it gets bigger in terms of size. But think of what that means. On an MRI, if, for example, I happen to be lucky, and I had one before and after I had depression, or during the time I had depression, and I saw that before I had depression, my amygdala was of a certain size, and it was bigger while I was experiencing depression, what does that mean? Why would it be bigger on an MRI scan? It's not just activity that's going up, but it's physically growing bigger in depressed individuals. Why? Can you think of a reason? What makes something bigger in the brain? Yes?
More neurons.
So I might have new neurons being generated here. I might have an increase in the arborization of the neurons that are there. So they just have, it's a really bad humid day and all the neurons, their hair goes poof. And that might make the neurons look a lot bigger. We don't know. It's a fascinating observation that is very consistent. The other thing that, again, is interesting, that the left prefrontal cortex very often, especially within psychology that's associated with positive mood, has both a decrease in size as well as in activity, kind of fitting along with the first bullet point up there. And again, the hypothalamus has been implicated. The HPA, hypothalamic pituitary adrenal gland axis, is overactive in some depressives, individuals, but not universal in all cases, just as a case in point. So overall, the brain is changing, but we don't see a consistent change. Some circuits are going down. Prefrontal cortical systems are going down. The Other circuits, the amygdala circuit, for example, seems to be going up. And so this means it's really inconsistent. We don't have a universal theory as to why this is going and changing this way. In Parkinson's, we see a loss of neurons. In Alzheimer's disease, we see a consistent loss of neurons across the entire spectrum. But here, we're seeing some things going up and some things going down. And again, from a neurobiological perspective, that should be driving you crazy as neurobiology students. Why are these structures bigger? Why are some smaller? The smaller part I could understand, but why are some parts actually bigger in some individuals? And I think now is actually a pretty good time to take a break. And if you can just hold on for 1 minute, we're going to switch slide decks.
Hi, everyone. A few of us in the course have gotten together in the past couple of weeks, and we've been writing these positivity cards that are meant to sort of just keep everyone in a good mind space and just remember to take care of yourselves. So we just kind of wrote messages that we would want to receive ourselves. So we'll just be passing those out right now. So I hope those even make the smallest little change in your day.
Nice, thank you. Thank you. So at any time, if you're ever not feeling okay, just let me know, and I will be trying to help you as much as I can. I mean that sincerely, and I know you're about to enter a really stressful time. If I can help you in any way, manage resources, if we can help with different HMB courses, just again, mention it, and we'll try our best to help wherever we can. And this is sort of the virtual hug that you're getting from your colleagues in this course. So one of the things that I'm hoping, if you are thinking about this, I told you very briefly that when we're looking at different treatments for depression, they don't all work. So if I give you an antidepressant medication, and I give you like Zoloft, or I give you some other drug, like a tricyclic antidepressant, it may work, it may not work. You'll have to go through another drug, it may work, it may not work. You might go through about 5 or 6 different drugs, and it may not help you, which is, again, really unfortunate. But we also know that some individuals are really resistant to any form of medication. Like, we can't help them. No matter what drug we give them, they are treatment resistant. So I wanted to talk about this one area. At the very bottom of the prefrontal cortex, in an area which is known as area CG25, and I'd like you to know area CG25, which stands for, the CG stands for the cingulate cortex. In the subgenual area of the prefrontal cortex, we actually have this small group of neurons that forms area CG25. Area CG25 is a really interesting area for us as neurobiologists. It's actually also really interesting clinically for people like Helen Mayberg, who was a U of T pharmacologist, and then she actually looked at this particular area. And one of the reasons why we're interested in this is that if we were to take a look at an individual who is, or just a normal individual, you all have an area CG25. If your area CG25 was working well, if you saw a really sad movie, or you heard a really sad song, or you happen to remember something really sad in your life, this area of the brain, area CG25, will start to become activated. And as it becomes activated, that sadness is sort of ramped up. So one of the things that makes this area CG25, unique is that it's chronically activated in some treatment-resistant depressed individuals. So these are individuals who have tried like every last medication out on the market. It doesn't help them. They still feel really, really sad. They are at risk for suicidality. We need to help them. And when we look at their fMRI scans of the brain or a PET scan, this area consistently comes up in these treatment-resistant individuals as always being turned on. It's like they're hearing a sad song in their head all the time. It's always being activated. So Helen Mayberg actually thought, well, in some individuals that have depression, that's treatment-resistant, if we can turn this area off, what might happen to those people? If we could somehow use stereotactic neurosurgery, so the surgeon will drill through the skull, implant an electrode surgically into only area CG25, and we use a technique which is known as DBS, or deep brain stimulation, and we turn off this area, what happens to those depressed individuals? And when they do this, not every individual, but 80% of those individuals who have failed on every other treatment regimen, they now feel that they don't have the depressed symptoms, and even better, they don't have a risk for suicidality. So again, we don't understand all of the biology that's associated with this, but it does tell us that there are different areas of the brain that when we can turn them off, if we were able to identify them, might be of some benefit. And this is an area of the brain that I'd like you to make sure that you know for the upcoming term test, in addition to the previous slide that outlined other areas of the brain that are affected. So this area, in some individuals, is overly active. It's not bigger in size, but its overall activity seems to be consistently on at high levels. We don't have a genetic model, so as a result, we can't take a gene, overexpress it, or knock out a gene to induce depression in animals. So what we have to do is come up with an induction model. We have to induce depression and then study the effects on the brain in these different animals. And there are a number of ways to do this, and I'd like you to be familiar with these models. I'm going to concentrate on the ones that are here in the middle. So there's the social defeat model. So if this mouse is exposed to a larger rat, then this mouse over time will actually exhibit signs of depression. And it's social defeat-mediated depression. We can also have this form of learned helplessness, where, for example, a mouse will learn to go into this chamber. The door opens. Normally it goes into this chamber. There's usually a reward associated with moving into this chamber. But to prevent that from happening, what we will do, or researchers will do, is to randomly shock the animal. It shocks the animal, and it doesn't know when the next shock is coming. What do you think the animal does as a result of having these random shocks delivered to it? Will it go into the next chamber? What do you think happens to it? It stops moving. It learns that no matter what I'm going to do, I get these random shocks, so basically I should just learn not to do anything. And this form of learned helplessness is a way to induce depression in mice models as well. We could also socially isolate these animals as well, and there's also forms of intracranial self-stimulation in certain areas that can induce depression. We can look to see, for example, that there are changes in behavior. So normally, if you were to take a normal mouse, and you were to expose it to a brand new mouse, it's curious, it's motivated by this curiosity, and will go up to see this mouse, and you can see the tracks that it would normally take. It stays on the outside of the chamber, but it spends a lot of time in front of this brand new mouse. If you take a mouse that has been socially defeated, though, and you were to expose it to a brand new mouse it's never encountered before, normally it should be really curious about going up and seeing what this new mouse is doing, but you can see that it actually doesn't. It spends most of the time kind of by itself. And so this model of developing the depression is actually really powerful. We also know that these are similar in many ways to the animal models that we developed for anxiety. So most people are now starting to lump depression and anxiety together. They seem to have a comorbidity. Often people who have depression also have anxiety, or people who have anxiety will also have things like depression. So again, if we're looking at these other animal models of helplessness, this might be a good sort of hint for you that maybe you want to think about these animal models as really short one-line answer questions. But there's this other model that people are now starting to look at with some more interest. So this other animal model is actually much more popular because it kind of doesn't model any one of these. It kind of looks at what is known as chronic unpredictable stress. And this chronic unpredictable stress is actually what we model here at U of T with undergraduate students. So once you start figuring out how things are going to be and you've adapted to it, we change the rules on you. So once you started figuring out how to answer multiple choice test questions, guess what? In third year, we switched so that you write short answer questions. Once you start getting good at that, we switch it again, so now you're doing group activities that you all don't enjoy. So this is sort of like chronic unpredictable stress. And what happens is, every day this animal is exposed to a different stress. So one day it might be sound, and it stresses the animal out. That sound is playing in the background, it can't go to sleep normally. And then the next day, instead of playing sound, you might actually start spraying water on the animal. So it can't have the ability to adapt. And this chronic unpredictability is a form of stress that will induce depression really easily. So I'm not going to talk to you about resilience today. We've spoken a little bit about resilience. It's actually a really fascinating topic, talking about resilience, but I'll leave that for another day. The last thing I'm going to ask you to make sure that you know, I've mentioned to you about anhedonia. Do you remember anhedonia? Yes? We need to be able to know if an animal still has anhedonia or not. Has it lost pleasure? And has it lost interest or pleasure, this anhedonia? If an antidepressant medication that we want to try out in an animal model is working, how do we know that the anhedonia has disappeared? Because anhedonia is a really powerful marker for depression. And one of these is what is known as sucrose preference. So animals like having sugared water or sweet tasting water, and usually they will drink this much more than if they're given a choice between sugared water or just normal water. They will prefer the sucrose-containing water much more. But we don't see that in depression. It's about the same. They don't care which one they're drinking. But we know with antidepressant medication, we can measure whether the anhedonia is disappearing because they start wanting to drink sucrose more. So again, a couple of other things. We need to also test, not just for the anhedonia, we actually have to test for motor effects. We need to have a test to see if social isolation worked or if chronic unpredictable stress worked and we induce depression. How do we know that the psychomotor effects have actually taken place? I've already told you about this tail suspension test, that if you hang a mouse upside down by its tail, normally if it's a normal mouse, it will struggle to upright itself, but a depressed mouse will simply hang there. It won't actually try. It will give up. It's already learned that it's not worth it. There's no struggle that's associated with it. I know some of you are thinking about U of T when I just said this, things about struggle, but it's true. But the mouse will simply just hang there. So these induced models are really important, but we need to test to see if depression actually works. And then there's this forced swim test. And these forced swim tests will also tell us a lot about whether an animal has depression or not. The foot shock models I'm going to leave out for the time being, so you don't have to worry about this. The tail suspension and the forced swim test as a way to go back and test for whether depression was induced are things that I'd like you to make sure that you know for the upcoming term test. So this is an example. I'm hoping it's going to work.
Switch to the mouse.
Switch to the mouse. From rat to mouse. Okay, awesome. Who says neurobiology is not comical?
And then click it.
So here you're going to see a forced swim test. And you can see that the animal is banded, so we can identify this rat. And you can see that it's trying to climb out of this chamber. This is showing no signs of depression. It's struggling. It's keeping up with the struggle. And now, after depression, it's floating. And this, again, shows that this is after inducing depression. We might have used like foot shock. We might have used chronic unpredictable stress. You can see there's a big difference. Again, normally an animal will try. It doesn't normally float. It tries to get out of the particular chamber, this forced swim chamber, and you can see that there's a very big, pronounced, easily defined difference in behavior. So these are the reasons why we have a model to induce it, but then also to test, did depression actually occur? Do you know, can you understand the differences between how we induce it and how we test for depression? Okay, so the two models for testing whether depression occurs is the forced swim test, which I've just shown you in this video, as well as the model of the tail suspension. If it stays immobile, then we know that it's still depressed. So ultimately, as neurobiologists, we have to be really concerned by the fact that we have no tests for things like depression. We kind of rely on a clinician sitting there talking to us about our symptoms, and this becomes, again, rather unappetizing because we know there's biology. And so individuals, for example, like John Downer, who's in psychology, have actually started to figure out that maybe we can use sort of more modern neurobiological imaging techniques that will allow us to separate out individuals that are going to respond to treatment or have different types of biology that's associated with them. And what they're doing is using what is known as resting state fMRI. You don't need to know all of this. I'm going to come back after the break, so it's just a prelude to the break. This part you don't need to know at all for the final, for the term test. But what this is basically saying is that based on the way the different rodents as well as different individuals respond to different types of treatment, the types of conditions that they exhibit, whether it's a lack of motivation, increased anxiety, it suggests that there are specific neural circuits that are involved. I'm going to try to relay that all to you today. And as a result of all of this, it suggests that we can actually examine these circuits in some more detail. And that's exactly what they did. So in Jonathan's paper, which I'll go through after the break, after the term test, he actually identifies 4 categories or 4 endophenotypes of depressed individuals. And these 4 biotypes or endophenotypes, using a new type of fMRI, which is known as resting state fMRI, actually, if it turns out to be replicated and it turns out to be elaborated by other groups, this is the first sort of diagnostic test. That if I have depression, or I suspect I have depression, I can walk in, ask for a resting fMRI test to see whether or not I fall into one of those 4 categories. If I do, I might be able to get a more definitive diagnosis of depression, and more surprisingly, if his results are correct, there might be ways to actually predict which biotypes will respond to RTMS, or transcranial magnetic stimulation, or to traditional antidepressant medications. Again, it's a really exciting paper that came out last year. It has to be replicated, it has to be elaborated by other groups, but if it's correct, this is the first sort of diagnostic test for depression that people will be able to understand. And it also gives us some ideas about how treatments will work. Because I can tell you firsthand that when you find a drug that doesn't work for you, and you have to go back 3 weeks later and I have no difference, like this drug actually makes me feel less motivated, please give me another prescription. And we try a different drug or a different dosage of a different drug. You might be doing that for 2 or 3 months and you want to get better, and that's a bad thing. And if this works, this gives you an inline to something that can maybe speed up the entire process for you so that you're not sitting as long in this depressed state. So that's something that we'll be looking at later, and these 4 biotypes are ones that we'll be looking at a little bit later, related to exercise, the impact of exercise on these circuits, if it actually alters these circuits at all, but we'll be coming back to this after the break. So I'm going to go through a review with you really quickly now. And if you have any questions related to any of the lectures, please let me know. And we're going to go through them lecture by lecture. This slide deck will also be made available to all of you as well. So you don't have to worry about trying to copy everything down. I'll give you the full slide deck as it's coming up. Is there...
Just streaming stuff. Okay.
And is there something that they can log into early?
They'll get a link once it gets
Okay.
Yeah.
Okay, so I'm going to give you this sort of midterm review, and then I want you to make sure that you are aware of certain things. So I want you in your first lecture, when we're going through that, I want you to, again, make sure that you are familiar with this slide, that's in the slide deck. Spend some time on it. And I want you to make sure that you have a clear understanding of, for example, the bacteroides, these gram-negative rods, and the firmicutes, these gram-positive rods. You don't really have to worry about knowing the differences between phyla, divisions, etc. But this slide, knowing the overall major players, is one that I'd like you to be familiar with. Some of you have been asking about that first pre-lecture checklist question. One of those questions actually was incorrect in terms of the response that you should have. I'll go back and correct that later. But this is a slide that I want you to also spend a little bit of time on. I'd like you to know the overall changes in diversity. The numbers on the right, as I mentioned at the time, you don't need to worry about the overall numbers, like 10 to the 2nd versus 10 to the 7th, etc. But I do want you to know that there's an increase in diversity. And I also want you to make sure that you have a good idea about the different types of bacteria, especially ones that we have seen several times on several different slides. So have you guys all studied this particular slide? Yes? No? Kind of? If you haven't, it's a good idea to make sure that you go back and study this slide. Again, this is another slide that I'd like you to be familiar with. This gets back into the bacteroidetes versus the firmicutes. There's always a balance. We end off with one of the diagrams where we start off with a 2-year-old having this balance between the bacteroidetes and these firmicutes, and then that stays consistent or relatively constant throughout the rest of the life. I'd like you to, again, think a little bit differently because you know you have some one-line answers coming up on your test. Then maybe there's a definition that you might need to be able to clearly indicate. Things like enterotypes. Would I be able to define it? It doesn't have to be the exact same wording, but could I define an enterotype for a question asking me to give a definition? Again, what is dysbiosis? It's another definition. Some of you have had immunology, some of you have not, but it's a definition that you should, again, have some familiarity with. And I was asked this earlier today, so I wanted to actually talk to you a little bit about this. I'm not going to ask you a question where, like, if you were to do X, like, how soon might you be able to see these changes? I mentioned to you that there is this real disconnect between doing animal studies and human studies. And I told you that the Wu et al. Paper in Science, there actually hasn't been a follow-up replication of that study done the way that they did. And this becomes a really big issue. Because think a little bit about what we've learned. If you were to think about doing a change using probiotics where everything's stacked in your favor, would you really be able to see the same type of change in gut microbiota now? Most people would argue no. Like, you take like a whole bunch of yogurt, you eat that yogurt, will it change enough of your microbiome tomorrow, 24 hours later? Most people would argue no, it won't. And so this is a controversial paper, but just learn it the way that was presented to you. And again, in mice models, it seems to be a little bit more consistent. So I want you to know terms like the meconium, because we talked about the meconium earlier on. And I want you to, again, make sure that you know all of the features that are normally associated with the meconium. And how many of you think that there will be some sort of question related to this diagram? Not all of you. Okay. So the rest of you should now start putting up your hands because I can tell you that there are several questions related to different aspects of this particular diagram. You need to go through it. If you haven't studied it before, make sure that you understand it. I'm not going to ask you to draw this if that's your concern, but you should know like when different things happen, if you are being born vaginally versus a cesarean section, like you should know the types of bacteria that you start off with. Everyone then gets back into the same track. We all then develop these bacteroidetes and these firmicutes that are in balance as our normal enterotype throughout most of the health. Again, this just reiterates that by the time of 1 to 2 years of age, that this is fairly stable right up until adulthood. And again, you don't have to worry about numbers. I'm not going to ask you like what what percentage of the viral sequence is it 56%, 60%, etc. I don't like those types of questions. Or if I want you to memorize a certain type of number, I'll indicate that for you, but that isn't one of them. So other things that students have been asking, like, do you actually have to know the individual papers by name? No, I'm not going to be asking you, like, which of the following was found in the Turnbaugh et al. 2006 paper. You don't have to know the names of the different studies. I don't think that that's of any use to you, to be quite honest. Like, it's not going to help you like 10 years from now. Yes, that 2006 Turnbaugh paper, I just can't put it down. It's fascinating. But other things, for example, changes in the bacteroidetes or increases in actinobacteria, that might be useful. So I also mentioned that you should know parts of this pathway, I think. I mentioned that to you. And again, I want to just confirm that you will likely have some iteration of the data on this slide, whether it's the anatomy, the pathways that are involved on your return test as well. I don't want there to be too many surprises. Having a few surprises is okay, but too many is actually not good for you in a space of 1 hour. Any other questions from lecture 1? Things that you want to know, you have to know for sure, don't need to know? Assuming that all of you have vigorously been studying lecture 1, fully prepared, ready to do it. Yes? No? Okay. So I'm guessing the next slide. Wait, how does this work? Okay. So if you can get into this website, so polleverywhere.com, HMB300 fall, and we'll just see, hopefully some of you have done this before. We used to use Poll Everywhere a long time ago. They keep changing the rules on us. If you don't know who these teams are, just take Golden State Warriors. You, 20% of you are picking Avon? Oh. Okay. All right, so I think we're in pretty good shape. Oh, no. Okay. We better move on before it reaches anything like in 30% range. Okay. So what grade do you think you're going to get on this turn test? I kind of like that. That's good. Okay, so I think we're all familiar with this polling now, so we'll get on to what actually... What? Okay. Change your answer. Okay, so this is actually a real question, one that I want you to think about. I just showed you that slide, by the way, so it should be fairly fresh in your minds. And if it isn't, think about this really, really carefully. Refer to your slide now. This is a good opportunity to make sure where you understand this question is coming from. Are we all happy with this? Yes? So lactobacillus is the one that's pretty consistent through every part of the digestive tract. What's the other thing that makes lactobacillus really important for us? Where do we get it? So where did we get that from the skin? So again, this one source was early on in development, vaginal delivery. The source was probably the skin. So that would be great. So we're at 100%. Awesome. Okay, so just moving on. Okay, I don't see it here, but I'm guessing that it's okay up there. So when we are looking at lecture 2, so each of these represents the different lectures, and you'll have 4 that you need to worry about. So different drugs appear to target different regions in the reward pathway. Make sure that you know those. And I think I indicated this to you earlier on to make sure that you go back and review this particular pathway. Please make sure that you are. There is 1 or 2 questions related to this slide and the accompanying slide that goes along with it. Make sure that you're spending time on it. I know for some of you it's a review, and for a very small proportion of you it's not. So for those small proportion where it's completely new, make sure that you're giving yourself enough time to know some of the different structures that are involved and some of the different drugs that are involved in activating some of the different cell types. Again, also make sure that you understand the progressive rewards model, what that actually represents, why we might use that in a certain setting, especially related to dynamic contrast or dynamic novelty, how we might actually use this. It's more likely to show up as either a short answer question or a multiple choice test question. It's not one of your short answer choices or the 5 or 6 line answer choices. It's more likely the 1 line responses that I indicated to you earlier. Again, also make sure that you are going through the different aspects of Parkinson's disease. I know, again, for many of you, you've had a lot of neurobiology, but for some of you, it's fairly new. So make sure that these background slides are ones that you're comfortable with. If you don't understand things on here, you don't know what alpha-synuclein is doing, please come and see me as soon as possible. And again, just making sure that in terms of the bullet points that I covered, that when I put bullet points up here, I may not have actually told you a lot about different types of diseases, but I do indicate that alpha-synuclein is not only found in Parkinson's disease. Alpha-synuclein is pathogenic to a number of different diseases, including Parkinson's, but also multiple system atrophy, Lewy body diseases, etc. So any of these bullet points, even if I haven't fully expanded on what is multiple system atrophy, you'll have to wait and see in some of your other third-year courses. I would still expect you to know that sort of data. So let's actually try this, because this is coming from the... And I think this is a later version of it, so I have to apologize. Let's see how you do first, and then I'll talk you through it. Okay, so Bill's worried that he's been infected with a prion-like substance after eating a cheeseburger during class. He immediately goes to the emergency room and explains the situation to the staff. Which of the following procedures does he unsuccessfully ask for in order to prevent the possible spread of prions into his brain? So we talked about different models in our context. Do you remember what this hypothesis was called? So Brack's hypothesis, if that's new to you, please go back, review Brack's hypothesis. And if you understand Brack's hypothesis, do any of you want to change your answers? And the one thing that might make the 3% and 1% or 4% and 2% actually change their minds. This year, we didn't really talk about the glossopharyngeal nerve. We only talked about the vagus nerve. But if I just cut my vagus, would that have helped? So normally, the transmission from the gut to the brain is thought to occur along the vagus nerve. So that would be the best answer that I was looking for. And that's the best answer that I was looking for. Okay, never mind. Okay, so cutting the vagus would have been the correct answer. Okay, so make sure that you know the different motor functions. Make sure you understand what happens when you have a specific pathogen-free mouse. Also, like what a germ-free mouse represents. How you can actually convert a SPF mouse, a specific pathogen-free, into a germ-free mouse. The Phi1 promoter, Phi1 alpha promoter, what its function is, what it normally marks, why we use it, all things that you should know from this particular slide. You should also know the behavioral tests that we looked at in terms of seeing a dysfunction in motor coordination. So again, this particular slide here, I've included it, not that you need to know the data, so you don't have to know and be able to tell me about the overall changes in density, etc., that's represented on these graphs. but I would expect you to know some of the technical details of how I would detect, for example, the aggregation of alpha-synuclein in areas of the brain like the caudate putamen or the substantia nigra. Why I'd be interested in the caudate putamen and substantia nigra, what the purpose of the phosphostyrene antibody for alpha-synuclein is, what neurotrace is, or what methylstaining is. So if those are stuff that you don't know right now or things that you aren't sure of, then please come and see me as soon as you can. And then, of course, making sure that you understand about solubility and the role of triton extraction in the soluble as well as the insoluble alpha-synuclein that accumulates within the brains of these different animals. So, again, from this, I had a lot of questions about this particular slide, and I wanted to just clarify it because I'm keeping it simple for your term test. If you have a normal resting microglial cell, a normal resting microglial cell will have a certain phenotype that's associated with it. And this will be things like having an elongated cell body as well as really branched arborizations. If it is activated, if there's inflammation going on, if there's some pathology associated with it, the microglia actually has a rounder, more amoeboid-shaped cell body. The processes become shorter. And this is, again, the basketball analogy. It's like looking at Kevin Durant being morphed into Kyle Lowry, right? So you get this really tall person with like long arms and limbs into a short, stouter person who's great at driving the lane, but really bad in the playoffs. So again, just to keep it in terms of the general shape changes, please make sure that you're aware of that. That's how I want you to know it. You should know that it can be marked or stained by the IBA1 antibody that is specific to microglia. You don't have to worry that I'm going to put up a picture, which I don't know what classes you've had before, but so many questions about, will you have a picture like this on your term test? And I said, why would I put a picture like this on my term test? Just knowing the general description in words of the changes is probably the best thing to do. Oops, I think I missed one. Please make sure that you study and understand that these activated microglia that have changed their morphology are also going to be upregulating, and that's one of the reasons their cell body becomes bigger and more rounded. They're upregulating the expression and the release of factors like TNF-alpha, interleukin-6, etc., the pro-inflammatory changes that we're observing, and make sure that you're aware of that. From this slide, again, as I mentioned, you don't have to worry about the details. I'd like you to know that we look for bacterial changes based on 16S RNA bacterial sequencing, and that normally we cluster these into operational taxonomic units or OTUs. If we see an increase in OTUs, we get unique, distinct 16S RNAs, more of them. If we see fewer OTUs, we're actually seeing a lack of diversity associated with them. So again, make sure that you know the general features of this. Don't have to memorize this slide at all. So this is a bit of a tricky question, which involves more than just section 3. So you need to know a few different things. So you need to remember what IBA1 is, and you have to also remember melanin, which I haven't talked about at all in this particular slide deck. But you do have to know that kind of detail. Okay, so we talked very briefly about area CG25 here today. Does area CG25 have melanin? Do you remember what area of the brain actually has like melanin, or what types of neurons have melanin? So those dopaminergic neurons, often associated with the substantia nigra, caudate putamen, most often substantia nigra, but this would be the best answer in terms of what I was expecting. The microglia clearly stained for IBA1, and then you have to differentiate. Would I see this more likely in the dopaminergic system or an area CG25, which is a forebrain bundle that she never talked about dopamine or any other type of neurotransmitter. And again, those are the types of level of difficulty type of questions that you should expect. I'm hoping that you'll be able to go through all of them really well. So again, from today's lecture, I'd like to just point out, stick to the biology. I talked about these different substances. You will see these substances again in different forms. It may not actually be the answer I'm looking for. It might just be a distractor. Make sure that you are comfortable with all of these different terms like isotretinoin and interferon alpha. Make sure you know the biology of the different endocrine systems, the glucocorticoid system, the thyroid system. Make sure that you know Cushing's disease, glucocorticoids, thyroid hormones in this Hashimoto's disease that you see here. Again, Cushing's syndrome, the major features there. Make sure that you know RU486 and its overall effects. And again, I'm going to be putting up a number of different drugs that are associated with these different pathways. Make sure that you can outline what iproniazid is or what reserpine is, etc. These are noradrenergic pathway drugs that were originally used in neurotransmitter discovery, but I might put them up there for a totally different question, just as a distractor. So make sure that you are clear that this is associated with noradrenaline and the depression system. And again, I talked about knowing this pathway. Please make sure that you spend time going over the biology of it. The slides that I'm showing you are actually ones that I've derived some questions from, so make sure that you spend time on it. I will post these immediately after the lecture today, so you don't have to worry that I'm going to leave any of those out. And we'll just do this last one. Really? No other systems? Oh, thank you. Awesome. Okay, so the answer I'm looking for here, which I think the majority of you have, is sort of the glucocorticoids. If I haven't done anything else to Jessica, then, or Jessica's mice, then we wouldn't have things like dexamethasone, etc. So again, make sure that you're good with that. Do you want any last-minute hints? So you should definitely know aspects of what Nissl staining is. You should definitely make sure that you know the differences in the genetic versus the models, the toxin-induced models of Parkinson's disease. So if you don't know any toxins right now that cause Parkinson's disease, it's probably a good idea to go back and review those. I'm always looking at different protein markers, so you should make sure that you know some of the different protein markers. Do you want anything else? And again, you'll have this slide, so you can try copying it down. I'll leave it up there for a while. I'm not giving you the parts that are underneath it, but this is going to be one of your multiple choice test questions. Wait for it. I promise you I'm going to post these for you, but if you feel like you need to take pictures of this, take pictures of me too. Okay. So there's one other thing. So I hate to ruin your Thanksgiving Monday, but on Monday we're also going to have an online Google Docs. You can ask questions in real time on the Google Docs if you have any questions. On Tuesday as well, I'll have extra office hours for you to come in. I posted the different room assignments for you. Please make sure, if you can, to get to the room that's assigned. Some of you are in MSB. Some of you are in Sid Smith. Some of you are in Wilson Hall. Make sure that you scope those rooms out. We're going to be starting at 5:15. You get the full 60 minutes, but you should be, as best as possible, trying to get there on time. Do you have any last-minute questions? Nothing? I will stick around. One last thing. Please have a fantastic Thanksgiving, and hopefully I'll see you all online on Monday.